Welcome, everyone, to another episode of Swarmstead Bees and Gardening Live. My name is Bruce Rodriguez. Tonight, my guest is Dr. Seth Coleman. I almost forgot already. He is a PhD in evolutionary biology, among many other things. I brought him on tonight so that we can talk about all kinds of things. We'll talk about how evolution, selection, selective breeding, artificial selection, how all these things happen in the animal and plant kingdom world and how they can relate to beekeeping. And I'll bring him in right now. Welcome. Thank you for coming on. I'm just really glad you were able to come in and talk to me about this subject. I've always really been interested in evolutionary biology. One of my favorites is Richard Dawkins. That's where I learned all my everything I learned about it. And so if you could, I usually just start by asking my guests to just give me a little background about yourself. Kind of what you studied and maybe a little bit of what you know about bees so far.
So I'm thank you so much for having me, honestly, first and foremost. And I'm as fascinated with bees and beekeeping as you may be with evolutionary biology and processes. So we'll learn from each other tonight, I think. So my name's Seth Coleman. I am from Washington State and just grew up with sort of a childhood love of like nature and the outdoors, and that evolved into wanting to answer questions that I didn't have the answers to, and almost all of them had to do with why some animal was doing something. And so that really just over the course of school and getting older and more focused, turned into a PhD in evolutionary biology. I studied the satin bowerbird over in Australia for my PhD thesis, so I got to spend a lot of time in the eucalyptus forests of northern New South Wales, which was incredible. But as we were talking, just a little bit before we came live, one of my favorite topics in the classes that I was taking in my dissertation had to do with the evolution of social behavior. And of course, bees are front and center when we talk about the evolution of sociality and social animals. And it's really cool. You have 20,000 species of bees, and they run the gamut from being totally solitary to hyper social, right? Like the honeybees that we're going to talk about a lot tonight. So I'm really glad to be here. This is a great topic. And thanks again for having me.
Okay, yeah, no problem. Thanks for coming. And so for full disclosure, are you a beekeeper?
No, I'm fascinated by it, but I've never kept bees. It's not something I know much about.
Yeah. So one thing I tell people is just that selection. First, can you kind of give me a couple of the differences between capital E evolution, which a lot of people say, and then how that's different from artificial selection by maybe some type of obstacle for an animal to survive, and selective breeding, which is more man-driven, right?
Right, and artificial selection is a term that's it means the same thing as selective breeding really anymore, and it just implies that it's not nature or natural pressures that are doing the selecting, but instead it's us. Selective breeding is just a more specific term because it really describes what it is we're doing. We're in charge of which animals get to breed or which plants in our population. Natural selection, and that actually is evolution. We use that when I'm teaching classes in biology or evolution. We use selective breeding as evidence for evolution, because quite literally what we're doing is we are choosing individuals from our populations and breeding those specific individuals because they have traits that are desirable, whatever that might be. And then those individuals, assuming those traits are heritable, that they have a genetic basis, pass on those traits and hence those behaviors to their offspring. And you end up with a population that has that trait of interest more widespread than the population before. That's how we, create everything from like taller, more robust strains of wheat that don't fall down in a windstorm to honeybees that produce higher yield or are more effective pollinators. Natural selection, so that is evolution, right? It's just literally seeing a change in gene frequencies over generations, and the genes in artificial selection or selective breeding are the ones we're trying to propagate. So we've identified that behavior and we're pushing that behavior forward. Under natural selection, sort of capital E evolution, we are not the selective agents. nature is, natural processes, predator-prey relationships. The wolf is driving the speed of the elk, and the speed of the elk and the endurance of the elk is driving social behavior and endurance in wolves. Because wolves that coordinate and cooperate better and have bigger lung capacity and can run further catch more elk, survive better, reproduce better than a pack of wolves that is less cooperative, less communicative, and less in less good shape, physical shape to track these elk down. And so you have, under natural selection, you have natural pressures driving the evolution of these populations, or in these populations. So obviously under that situation, we are not the coming in and quite literally saying, male A mates with female B because we want trait X in the next generation. That's up to nature. That's up to the speed of the elk and the speed of the wolf. So I hope that, I don't know if that kind of covered it, but yeah, it's certain traits being promoted in the subsequent generations. It's really just, the only difference is who's doing the selecting, nature or us.
Right. So sometimes some of my friends, even people who also do treatment-free beekeeping, which you'll get to know what that means.
Yeah, cool.
They'll say, I'd like to just get some bees and let evolution take its course, but that's not actually what it is, because once you got your hands in there poking around, you may be purposely or unknowingly selecting from many things that would not happen all alone in the woods by themselves. So you can't really have evolution happen in your yard. You're having selective breeding, kind of, or it could kind of be artificial selection. The examples that come to my mind are, have you ever seen those sheep that live on those really steep walls of that? Was it a bridge or something that they made?
They're on the stones of the bridge, right? They just use the bridge as though it's the cliffs, the natural cliffs that they're used to.
Okay, yeah. So, but those are like really, they're really steep, and the way they just run on them, and there's predators that can chase them, but they're constantly battling. But some of those sheep are going to fall down occasionally and die, right? Because if they didn't, and someone might see a video of one of those, I think it's a bighorn, whatever it is, they'll see one tumbling down and they'll say, oh no, they need to like build some type of thing so that when they fall they won't die, or they'll put some kind of extra grip on there,
like a sheep catcher at the bottom of every mountain.
And I try to explain to them, like, that's part of selection. There will never be a day when all the sheep will never fall again. Like, they will die. Something's going to die just to keep things going. And that's kind of how it is with beekeeping and how I keep bees. I don't treat not a lot, but a good number do die. But then a good number do survive. So, and then there was also the example of there were some moths that I forget what area it was, but they were white, but everything they landed on, they stood out really bad.
Great example.
And there was some type of smoke or something. I think it was the Industrial Revolution that caused everything to be covered in soot. So over time, they adapted a darker color where the white ones would just get smashed, but any little bit of blackness that would blend them in kind of helped them live. How long did off the top of your head, how long would that have taken?
So that is an incredible natural experiment that, like you said, occurred around the Industrial Revolution. These are the peppered moths of England. And initially, these populations, and to answer your question, just before we get into a little story, I think this occurred it was incredibly quick because of the strength of selection against either being light colored or dark colored on your background. And so I think that the transition from the population being predominantly white to predominantly black moths was really rapid, like 5 or 10 years of this soot. Yeah, and so and I love it that you brought up that story. And
I thought you were going to say 100 years, maybe.
No, it wasn't even remotely that long. Both because moths can have multiple generations, right? So they're not like us, where you're waiting 9 months, between pregnancy and giving birth and all of that. But the selective pressure was so strong because literally once these trees. So the story goes, and I love it that you know this one, but so the way that the story played out was, as all of these industrial factories come online, they start belching soot, into the air, and this soot coats the trees in the nearby forests that were traditionally light gray bark in color. And so light gray, kind of white, pretty bright colored bark. And so most of the moths in these peppered moth populations were light colored, because being a light colored moth against a light colored background made you better camouflaged. And if you were a dark moth, just happen to be, unfortunately, have that gene that you're a dark moth in a light colored world, you get eaten real fast by the birds that are looking for food. Well, as the soot starts to cover these tree trunks, all of a sudden, these white moths that were loving being camouflaged forever were all of a sudden standing out against the background of the trees. And now the dark colored moths that are sitting on these trees are the ones that are blending in. So all of a sudden, the birds, just being visually orienting predators, are like, they don't care. They're like, hey, that's a moth. That's a moth. And they just start eating the heck out of the white colored moths, leaving almost nothing but the dark colored moths. So the dark colored moths are the ones that are breeding, surviving, and reproducing, producing more dark colored moths. Now, what's really interesting is that after the initial sort of pollution pulse of the Industrial Revolution, England and a lot of other countries started taking steps to clean up the stuff that was, being spewed out of these big power plants and industrial factories. And so naturally, the forest started to revert back to light colored bark trees, predominantly light colored bark trees. And so you have this total reversal then back to the original condition. And we can actually watch this happen. And scientists did watch this happen in real time by collecting data weekly for years on the frequency of light versus dark colored moths. And what you see is that as the trees get dark, being a white moth is no longer beneficial. White moths are eaten, dark moths reproduce and pass on those genes for dark coloration. But then as the trees go back to more light colored bark, now all of a sudden those dark colored moths that were favored are the ones that are predominantly eaten, and the light colored moths now are favored. And now in English forests, it's rare to see a dark colored moth, because most of the forests are relatively clean, at least the color of the bark. And so most of the peppered moths are the white morph moth. So yeah, that's just too cool. I love that story. It's a really cool, it's a great story, because it's actually one of those examples where we see evolution actually happening before our eyes, over the course of a couple decades,
Yeah. So,
neat stuff.
one assertion that gets said to me very often is, you can't select for survival of varroa mites, which is the main pest here in many places. It's the varroa mites and their viruses that they carry. I mean, mostly the viruses, but they'll say the bees cannot be selected for survival for any of these things, because it takes eons of selection for that to happen. And an example like that, where an organism can completely change its color in a short amount of time, kind of contradicts that. And
it really does. Evolution can happen on really vast time scales, but it can also happen really quickly, right? I mean, literally, we, with my students, we can do experiments where we can kill, 90% of the bacteria in a petri dish, and then ask questions about those two colonies, that 10%, that survived, ask questions about why they may have survived. If we then allowed that to kind of, continue and just perpetuated those particular colonies that survived, and then tested them again and found that those colonies gave rise to are also resistant to whatever chemical application we just gave them. I mean, that's evolution, right? We've literally gone from a population in a dish that is 90% susceptible to a particular chemical treatment to a population in a dish over just, a few days, literally in the incubator, that's 90% resistant to that chemical, Right. So it's important to recognize that evolution happens on all sorts of scales. And sometimes we can quite literally watch it, when it comes to like bacteria, or like the peppered moth example, over, a few decades.
Right.
But it's also happening over eons too, right? So we have this huge time scale, and people sometimes get caught up in thinking evolution can only happen over these huge time scales. But if the selective pressure is strong enough, evolution can happen at any scale.
Yeah, I forget what the term was. I think it was, oh man, now I forget his name, but I watched the video. It was something like emergency selection or something when a
Hmm.
very drastic situation happens to a population. But um, so in the beekeeping community, there's the arms race between varroa mites and honeybees.
Right.
So going by what you said, a common practice in beekeeping is to fight these varroa mites, to fight them, tooth and nail. So there are certain treatments that you can use to knock your mite population down, like to 99% down. So you're taking
Right.
thousands and thousands of mites and turning them into a small little handful, a dozen,
Right.
repeatedly and repeatedly over and over again. How does what will be the outcome of something like that over time? I mean,
Yeah. So without knowing, like, I may not have the right answer here simply because I may not understand bee like generation times well enough, because obviously that plays a huge role in like the ability to evolve,
yeah,
right? Is
that's what I'm sorry.
bacteria generation times
Yeah,
can be 20 minutes, or a couple hours.
right.
um, but in your example, so my concern, and we see this in sort of industrial scale agriculture, whether it's crops or whether it's domesticated animals, that the more inputs we have in the system, yeah, it may keep your wheat or your cows or your bees alive for another season, but the more we assist with the survival of the organisms that we're trying to promote, the more risk we run of promoting organisms, colonies, fields that under natural conditions maybe shouldn't survive. And I'm not talking about like shouldn't, like capital S, like they don't need, they shouldn't exist. No, it's
Yeah,
more like they don't have the natural capacity to resist the stuff that's being thrown at them, like these mites, or bacterial infections in bacteria in cattle populations, domestic cattle. The more inputs we have to assist in that survival part of our organisms, the larger risk we run of really just stripping them of their natural ability to fight anything. We're producing sort of a weaker animal, when compared to like the natural state, or what we call sometimes the wild state or the wild type. So that would be my concern, right? Is if we're doing all the work to help these organisms survive that otherwise wouldn't. That's the key, right? These that particular bee colony or some cattle or some corn wouldn't survive if we didn't throw a bunch of chemical treatments at them. That's great. We can send that honey or that corn or that beef to market that year. We don't maybe lose out on the financial costs of them dying and not going to market, but in the long run, we're certainly not doing the population any favors in terms of like their ability to fight, like mites or bacterial infections. And that would be my concern.
yeah. The one question that was asked to me, they were kind of quizzing me. They said, what are some of the things that determine whether an organism can be selected for something? And
Hmm,
off the top of my head, I just thought lifespan, like you said, whether fruit flies can adapt much quicker than Galapagos turtles could to a
bingo.
drastic event.
Yep.
Um, Honeybees can reproduce very rapidly. Like one colony
Yep.
can turn into 4, 5, 6 in one season
Right.
easily. Um,
Right.
and varroa mites also reproduce very rapidly,
Hmm.
just very rapidly. I don't really do the testing and the treating or anything, so I don't know all those facts right offhand. But
Yeah.
yeah, so they do. And just as you were saying, not only are you knocking down 99% of the mites, and man, they're stronger, but the bees totally don't do anything for themselves. So if you weren't there for them, they would die. So what I would want to do is kind of flip it around the other way. So,
Hmm.
and it's kind of what I'm doing. I let the bees get knocked down as
Right.
bad as they can, or just as bad as it happens. Just however it happens, I accept whatever happens. So the bees get knocked down pretty bad, but they always seem to come back each season a little bit better, just with
Hmm.
no assistance whatsoever. So that's kind of the rift I was telling you about between uh, two factions of beekeeping.
Right. Right. And I mean, it makes some sense, and we see this in other forms of agriculture. And when we're dealing with domesticated animals, that the larger scale the production, typically, the more non-natural, like chemical inputs are forced into the system to protect the organisms we're trying to raise and get to market. Um, and yeah, and so smaller operations tend to be able to sustain themselves without the really large chemical inputs. But as you might be able to speak to, that's a pretty work intensive. Even though it doesn't require you going out and chemically applying anything, it's a fairly work intensive sort of way to farm. I have some friends who are organic farmers. So they just work on really small plots of land without any chemical inputs, obviously, and that they find it really rewarding. But it's also a ton of work, right? Just a ton of physical labor that you in order to protect their crops, that in a larger operation is largely done by automated spray machines, right, that are applying chemicals to remove the aphids, right, as opposed to maybe biological controls like ladybugs, or physical removal by like shaking the plants down or something. So I can understand, I understand the motivation, I think, to use chemical inputs to try to enhance or promote the survival of your crop or your organism. At the same time, like I said, the concern is that the more we are applying chemical inputs, the more we may be compromising the organism's natural or innate ability to fight anything, and so that I think is concerning. Also, kind of like you and I were chatting a little bit before the live started, one of the problems that we see with the widespread application of chemical inputs like pesticides, fertilizers, herbicides. I'm thinking more in like agriculture, but certainly it's probably pesticides that are being sprayed on the applied to bees, right? Because they're trying to kill the mites, so it's some form of a pesticide, probably.
Well, I mean, technically, it kills a pest, so right, so it'd be a pesticide,
Yeah,
but
some sort
they're
of pesticide.
usually organic acids, so they use
Okay.
formic acid, oxalic acid, different kinds of acids. There's also some really what would you call them? I don't even know, just chemicals like really bad that and then there's also people using like illegal things or at
Yeah.
doses that are not accepted by the
Right.
laws and
Or like the
everything.
EPA. And that, yeah, you actually hit the nail on the head. That's kind of where I was like going with that, is that what we see in systems, agricultural systems where chemicals are widely used, whether they're pesticides, herbicides, is that again we get back to this 99% problem, or I guess it's the 1% problem, right? Where you spray your field and you kill 99% of the Johnson grass or the invasive weed that's infecting your corn or your wheat field, and you kill 99% of that Johnson grass. Great, you get a decent wheat yield from that year. But 1% of that Johnson grass that didn't die is now going to seed in your field, and those seeds are going to sprout into little Johnson grass weeds that you can't kill with the chemicals you've been using because they are resistant. They are born of resistant parents and they inherited their genes for resistance. So now the farmer is stuck because now, like you were saying, they either have to up the doses or they have to go to another chemical. And of course, the danger of the second, third, fourth, fifth chemical is that the first chemical is the first for a reason, right? The first chemical on the list, like to use, is always going to be the one that's the most benign, the least harmful to other organisms in the environment, to the groundwater, to humans that consume it. But as resistance evolves in your population of pests or weeds, whatever they might be, you have to either increase your dosage of that chemical, sometimes pushing up against like the EPA guidelines, or you have to switch to a harsher chemical, one that's not as friendly to the environment or human health or the other organisms in your area. So yeah, that's one of the big dangers, right? Is like once we start that application of chemicals, it's where do you stop?
Well,
Because again, because of selection of the pests, for resistance. You know?
So I have a question. There's a couple questions, some good comments coming up, but we'll get to those
Sure.
when I'm done. But
Yeah, no worries at
yeah.
all. This is fun.
So one thing that is commonly said to me. Any of the beekeepers here that watch this will have heard it, will have read it. Beekeepers asserting that this oxalic acid treatment for varroa mites cannot be selected for. Like the mites will never become resistant to it. What they say. It's very common to say that, but you probably don't know this. But maybe 12 years ago, 13 years ago, when I started, they were using only, I think it was a gram of oxalic acid like once a year. Like that was it.
Right.
And it was only if you found 30 mites per 300 on a mite test,
Right.
on a mite count. And then it progressed to where currently they treat basically no matter what, but any even 3 mites in a mite count is the threshold. And
Mm.
they've gone to where they're using 4 grams per per box. It's kind of like beekeeping jargon here, but and they do this multiple times several days apart and then do that whole system several times during the year. So so it's kind of a loaded question, but if something goes from being used once a year in a small amount to dozens of times per year in large amounts, what are the limits of something you could be selected for? Does it ever make sense to say this bug cannot become resistant to this thing?
No, it really doesn't. And I don't know enough about the chemistry of oxalic acid and what it does to the physiology of the bees or the mites to be able to say much about that. And that might be important information, that would help us answer this question. At the same time, you're absolutely right. Like seeing that kind of increase in the dosage needed in a chemical input typically is like the signature of resistance evolving in your system, and you're only seeing the same results or the desired results by increasing the dose, right? So I don't know that that's what's happening, but that it sure suggests that that's what's happening in these mites. And also, I mean, if this does the oxalic acid kill the mites? Does
Well,
it
I'm
actually
glad you asked
kill?
that
Yeah.
because um, what I was going to say is nobody really knows exactly how it affects mites. They just
Huh.
know that when it's sublimated, or commonly they say vaporized, it's heated to a high temperature and then it becomes a cloud of pure oxalic acid gas inside the hive.
Okay.
It
Okay.
seems to not harm bees,
Yeah.
but after this is done, mites fall. They
Hmm.
fall and die on the bottom board. More beekeeping terminology, but nobody really can answer how oxalic acid kills mites. They'll say that it
That's
burns
interesting.
their soft tissues, but
Yeah.
that has not been proven. Like that's just some people joke and they say it burns their face off, but
Yeah.
the fact is they do not know how it affects them. It just does.
Yeah.
So
That's
we
really, I
really
mean,
don't know.
if it's killing them, and I don't want to sound silly, like, anything that kills you, you can evolve resistance to. I mean, I'm not going to evolve a resistance to bullets anytime soon, so I'm not like going there that just because something kills you, you can evolve resistance to it. But a chemical that's applied, that is applied to a system and it kills a certain number of animals. I mean, that's something that's interacting with their physiology. I don't see any reason. I don't see any way you could say mites could never evolve to this chemical exposure. I mean, it's a chemical, it's interacting with their physiology and it's killing them, but not all of them. So, again, I don't know how you could make the claim again, without doing some pretty darn rigorous studies, and they'd have to be longitudinal, right? Like across multiple mite generations to really be able to say with any reassurance that mites either can't, but even that I wouldn't. Most researchers, scientists would never say something like this animal simply cannot evolve resistance to this chemical, because that just doesn't make a lot of sense. It's a chemical interacting with the physiology. There are so many ways an animal's physiology can adapt to its environment, that I I strongly question, without wanting to throw myself into the center of this
Yeah,
debate.
yeah, I'm trying. Yeah,
I strongly question any claim that this chemical that's killing all of these animals um, is impossible for the animals to evolve resistance to. It just, that kind of flies in the face of what we understand about the evolution of resistance to chemicals and herbicides, pesticides, what have you. So
yeah. I mean,
kind
I
of
think of those. Somebody said something about it's so hot, or it'll burn you. You can't adapt or coexist with something that burns you. But I was like, we have crustaceans that live in volcanic vents in the ocean that where the water is like several hundred degrees, and they still live. So I
well.
don't know about
And are
that.
they? Does the oxalic? You were kind of saying, we don't totally know how the oxalic acid kills the mites, but it doesn't kill them because it's hot, right? It's
Right.
interacting somehow, or is it actually the temperature? Does it go in as a
No,
as a as a vapor, like a superheated vapor?
it's heated on a really. There's several different gadgets now that vaporize, or yeah, vaporize the powdered oxalic acid. But
Mm.
once it's inside the hive, it's not hot.
Okay,
It
yeah,
just flows out of every crevice and crack. So they kind of try to block all the air in there so that nothing can get in or out. It doesn't
sure.
seem to bother the bees. So yeah, it's not hot. But I think what
So
they
it's not
meant
a
by
temperature
burn,
thing. Yeah,
they meant like acid burn. But I was just saying,
yeah.
using the heat as an example. Um,
Yeah, and maybe if it's just like a caustic reaction, that the pH is at like the perfect place to burn mites but not burn bees, I suppose. Again, I still wouldn't be comfortable saying they couldn't evolve some resistance to it. Um, you know, so
and,
anyway, yeah,
yeah, and also a little more history of these treatments for bees is there have been many, many treatments that are all absolutely worthless now. I
mm,
think they kind of like reintroduce a couple once in a while, but there's some that are just totally pointless. They don't work at all, even though they did work very, very well when they were first introduced.
yeah,
Um,
interesting. Yeah, and yeah, that has happened repeatedly in agriculture. That not fertilizers so much, but herbicides and pesticides that used to work are either don't work at all or are much less effective now. And again, that has everything to do with evolution of the pests that we're trying to kill with them.
right. So you were talking about studies, and that was another question I had. Well, here let's look at a couple of these first.
Yeah.
Muller Hellrat says, I think this is more of just a comment. Says it also sounds similar to how
Yeah.
antibiotics can compromise the gut microbiome. They kill everything rather than targeting a specific microbe, so killing all beneficial organisms.
Yeah, absolutely. There are parallels that run between that and what is happening in bees. Certainly, some of the early applications before the EPA really stepped in and said, "Hang on, we need to evaluate how these chemicals are affecting non-target insect species." But before that, in the early 1900s all the way up through the 50s, 60s, we would apply just broad scale pretty harsh chemicals to our a lot of our agricultural fields that would kill not only the target pest moth and its larva, but would also kill the monarch butterflies that are feeding on the milkweed next door and the honeybees that are pollinating the fields all around, or even the crops themselves. And so yeah, that's a great analogy using antibiotics. And get slightly off topic, but the broad, really rampant over prescription of antibiotics that has sort of been traditionally characterized sort of Western medicine, at least in the United States for sure, has led to massive problems with people's actual gut biome. Because as kids, I'm a product of the 80s. I feel like in the 80s growing up, we were just the test subjects for everything. We had the red M&Ms before, like red dye number 5 was banned because it was causing cancer. There was something in the Doritos and the Pepsi when I was a kid. It's just like they were just trying everything on us until a substance would be banned, and then we'd just be
Yeah.
like, okay, the red M&Ms are missing.
Yeah, I don't know how I'm still even alive today
I
after listening
know.
to this.
I totally agree. Like, I don't know how I got through it. I'd like to say that I'm stronger for it, but I'm not so sure. I think some of that stuff does damage. Um, but it's. I also grew up, at a time when it was almost just. It seemed like doctors would throw antibiotics at just about anything, and that the result of taking those broad spectrum antibiotics, like Muller-Hellrat was pointing out, is that you end up killing all of the beneficial bacteria that are such an absolutely essential part of our gut microbiome, our ability both to maintain like a balanced physiology as well as extract maximum nutrients from what we're eating. All those beneficial bacteria die every time we take a broad scale antibiotic for an infection that most of the time had nothing to do with our gut, right? It's just
Yeah.
that broad scale antibiotic was going to kill any bacteria because it was interacting with their cell walls. So any bacteria in your body that came into contact with that molecule was going to be destroyed. And similarly, that was kind of the approach that we took, that kind of shotgun approach. Let's just take something nasty and throw it at the field because it's going to kill those moths and their larvae. That was kind of the initial approach we took to agriculture and agricultural pests. And we're still kind of recovering from that in a lot of states, honestly, a lot of big ag states.
Yeah, so that made me think of something. A lot of people in the beekeeping community, they have this kind of fantasy that someday there will just be no more varroa mites. Somebody is going to figure out the thing that kills every single varroa mite in the United States. And what I would say is, okay, for how long? Because they got here on some shipping container, they'll just be right back, in a couple of
Yeah.
years. Um, so do you know offhand if we've ever caused a pest to become extinct in the United States? I mean, I couldn't think of one. That
A
might be kind of
pest.
off subject.
Have we ever? No, it's not off subject at all. Have we ever driven the extinction of a pest? I want to say that we have, but I honestly can't think of a good example. Um,
Somebody said there was a weevil, but then I don't think the weevil was driven completely off, right?
Yeah, I don't think so either. And then you can talk about, like the European corn borer. That's that little moth and its larva that, like the name implies, bores into the stalks of corn, causing them to fall down, and you lose all your corn. I know that we introduced. That's one of those really neat examples of genetically modified organisms, where we genetically modified the corn to contain a protein that, when consumed by the larva, caused a blockage in the larva's digestive tract, and the larva died and never metamorphosed into a moth. If you never become a moth, you can't reproduce. And I know that was hyper effective against European corn borers, but whether they were driven to extinction, I don't know. I kind of doubt, because they can switch to other plants. They just preferred the corn for all sorts of reasons. Monocultures are easier to attack because every single stalk is the same. Every single plant has a really, really super similar physiology. And so they're more susceptible to a single pathogen or pest than like a more diverse field, So I don't know that. The short answer is I don't know whether we've actually driven any pests to extinction. I kind of don't think so. Yeah.
And what you were saying about killing non-target species, I think that, it's just something that I think. I don't know for sure, but if there was a predator for these tiny parasites, like parasites probably also have parasites in some
They
cases,
do.
or they have predators. So there could be something that could be fighting them if we could just kind of let things alone also, but going in and wiping everything out kind of just keeps restarting everything.
Yeah, I mean, and that's a great point. There really are so much more like sort of scientifically creative ways and more natural ways to promote the success and survival of your organism of interest. For example, some of the things I was reading in just prep for our discussion today, there are some really neat anti-mite behaviors that we see in both honeybees and in their really close relatives. I forget, it's another apis species actually found in sort of northeastern Russia. And it's actually where the mite came from. So the mite is native to Siberia, parts of Siberia, and there's a particular bee species, and another Apis bee, same genus as the honeybee, that is also native there and has evolved with this mite. And the mite still causes problems. It causes the failure of some colonies, but they've evolved to live with the mite, and they have these really cool behaviors related to getting rid of mite infection. So what they find is that this other species is really effective at identifying the chemical cues of larvae that are infected with the mite already, where the mite has already laid eggs on the larva. And what this species will do is they are hyper effective at either removing larvae, or they'll do something called recapping. And they'll open, quite literally, they'll open up the cap of the little larval tube. I don't know if I'm using the right terminology, honestly, but a little larval tube, little like, the little space where the larvae hang out. They'll basically open it up, smell it to confirm, yeah, this is infected, and then they'll recap it. And any mites that emerge from that larva, and if there's a female in there still laying or just hanging out with the larva, female mite, she and all of her eggs, or the larvae that have hatched from her eggs, will just simply literally be sealed and die in that little chamber. Also, these bees practice grooming. They're hyper vigilant about grooming themselves and others. So they do a lot of what's called allo grooming, which in primates, it's a really social behavior. You see like little like trains of chimps just kind of sitting on a log, and they've all got their backs to each other, and they're just, this one's picking ticks and stuff and fleas off the one in front of it and eating them, right? That's a little protein. And the one behind it is picking ticks and stuff off of it. And so they're grooming each other to get rid of parasites. Well, these bees are doing the same thing. They're actually grooming each other to help get rid of mites. And these are the kind of behaviors you see in species that have evolved with pests and parasites, right? They've survived this long, so they must have some ways to keep themselves from going extinct as a result of being exposed to this mite. The problem, of course, when you have an introduced pest like the mite in honeybees, because it was brought from Siberia to Moscow, and then Moscow, it ended up, like you were saying, in the United States in, I think, the 50s, like not until the 80s, wasn't it? It was pretty late.
Like the mid to late 80s.
Yeah, pretty late that it got.
What they say.
Right. Yeah.
I don't know how you can know exactly when they got here, but yeah, I think you're talking about Apis ceranae. That's the Japanese honeybee.
Yes,
or the Asian honeybee.
That's the one. So, but one of the really cool studies, there's a whole like series of studies now trying to figure out like how we can promote those kind of behaviors in honeybees, right? And so researchers are now looking at honeybee behavior really closely and asking questions like, okay, so if we observe a hive, a honeybee colony, where we see more of these behaviors like that removal behavior, or I think recapping has actually been observed in a few honeybee populations, grooming behavior, can we promote those behaviors by promoting that hive, right? Kind of like you were saying, you can, divide a hive, creating multiple colonies, and if, as long as those behaviors are genetically based and can be passed on to future generations, can we simply create honeybees that have more of these natural defenses, including these behavioral defenses, to this introduced pest? So there's some really neat work, I think, being done that doesn't require, kind of just falling back on chemical inputs, but allowing natural defensive behaviors to be promoted by us. Again, this is selective breeding. We're the ones saying these are the traits we want to promote. So these are the colonies we're going to really try to bolster, right? Divide and promote. Yeah.
So that comes up a lot in conversations, especially for beekeepers who don't treat there are breeders who select for that, what they call Varroa sensitive hygiene. So they do all those behaviors. So they really, really select for those things very closely. But my position would be that when you select purposely for one trait, kind of arbitrarily, these bees may do that thing really good, but are there other traits that we don't really notice are actually helping that are so for me, I don't select for any traits at all. I've had this question like it was the gotcha question for me a couple weeks ago. And I said I don't select for any traits. The only criteria for my bees is live. Like you need to
Right.
live without my help. So maybe it does take a little longer. I mean, I noticed a drastic increase in survival after three to four seasons. Um,
Interesting.
and that's what a lot of people find that do this, depending on what you're starting with. If you're starting with bees that you captured that already lived on their own, or if you're
Right.
starting with some bees that are coming from some bee breeder who pollinates almonds and just kind
Yeah.
of like a puppy mill for bees, so you
Right.
get these barely alive bees.
Huh.
Um, so here, there's you'll be interested in this one. There's they're testing extended release oxalic dribble on sponges and shop towels, letting it sit there for there's a long from what I heard, it's a long period of time. So to me, I'll see if you agree. Extended exposure to a non-lethal non-lethal levels of pesticides for a long time. Maybe it's lethal to a point, but what do you think of this long-term extended exposure of this chemical to try to kill a parasite? Um, that almost to me that would seem like that would lead to resistance even faster. Like,
Well, I don't know. It would be helpful if I could picture like where these towels are, but I'm assuming that the mites are exposed to these towels. The mites are already there in the colony or in the hive. You apply an oxalic acid treatment. You put these towels, maybe outside, and perhaps what's happening. I don't think it would to get to your question just real quick though. I don't think it would necessarily affect like the rate of the evolution of resistance, but I could see it potentially being effective again, depending on how the mites are getting to the colony and whether having like a towel soaked in oxalic acid around the colony, around the hive, would repel mites from even trying to enter. That seems to me to be a potentially, if they've got this towel, like basically a desert they have to cross, and it's pretty unlikely they're going to survive once they start out on this journey. You'll get fewer mites entering the hive. So I don't think necessarily it would speed up or slow down the evolution of resistance. Again, it kind of depends on how severe that selective culling is. Again, are you killing 99% of your mites, or are you killing most of the mites, but then you're doing a really good job by having these essentially like these oxalic acid moats or deserts around,
well, here
that you're just
I'll
not
help you.
you're not
I'll
getting more mites.
Yeah, I'll try to make it clear. I keep forgetting that you didn't, you haven't done beekeeping, so
Yeah,
you
I
don't
don't.
know.
I can picture a beehive, and that's about it.
So no, the way that mites do get into your hives, into your hives, they're brought in by the bees themselves. They
That's kind
go
of what I
to other
figured.
infested colonies. They
Right,
attach to their bodies, and then bees sometimes will go into other colonies. Sometimes they rob each other. Um, so the mites actually don't really survive very long on their own, just kind of walking around. Sometimes they get
right.
past. They'll detach on a flower, kind of like the way a tick would, and then the next bee that goes there, they'll grab onto them. So I guess that's how they diversify their genetics, by kind of going from colony to colony. That's
Yeah,
how they
absolutely.
tell
In
them
fact,
in.
one of the things I did read was that bee colonies in panmictic populations. So panmictic simply refers to the ability to mate with pretty much anybody else. And obviously, bees have a very specific mating system with, one or just a couple of. Are all honeybee hives single queen? Is it a single queen all species?
The majority of the time there's one
Yeah.
queen, but it's very common to have maybe two queens.
Okay.
Not often, much more than that, but
Okay.
there are several reasons why that can happen. Sometimes
Yeah.
there might be like a daughter queen that didn't take off and they kind of still get along. Sometimes there's very weird reasons why that can happen,
Right.
but it does happen.
Interesting. Yeah.
Um,
But it's a yeah. So that's good. It's good to know. But yeah, basically, what I found in some of my reading was that the more freely honeybees can mate with individuals from other colonies, the better they are at surviving mite infestation. And again, that probably has something to do with outbreeding, genetic diversity. by having a more diverse set of genes. Yes, some individuals in a colony might be susceptible, but all of these others aren't. And so it's interesting when we then bring in sort of the idea of like breeding systems, and how are we allowing our bees to move among hives and mate with sort of who they want, who they choose, to promote that sort of panmictic, more robust response to then like subsequent mite infection. It seems like the more inbred, and it's not surprising to any of us, right? The more inbred a colony or a population, if you just have colonies, around a small number of colonies around your field, or if you're forcing a single queen to produce most of the offspring for multiple hives, you may run into an inbreeding sort of problem, or just simply a problem where if all of your hives have the same genetics, more or less, because they're coming from the same queen, they are all going to be more or less equally susceptible to that next strain of bacteria or that next mite infection. The more diverse your colonies, or the more diverse the organisms in your population, the more it's a moving target for pests and pathogens.
Yeah, you get that shuffling of the
Yeah,
genetics.
exactly.
But I mean, to make it even more crazy is, a queen goes out and mates with with a large number of drones who could be from
Right.
anywhere. They could be from colonies miles away. So that I've often, wondered how inbreeding could happen. I guess it could happen, but I think, yeah, there's just so much genetic information out there with all the different colonies. Even if you have one queen and you make 20 daughters of her, by the time they get mated out in the wild, because they fly out and mate with the
Right.
feral pollination population.
Right.
They're your neighbors' bees, so it just gets shuffled constantly. But I guess it could happen.
Yeah, I just from the studies I read, just that allowing for this, and it sounds like that's the natural condition, this panmixia, where individual queens are free and drones are free to sort of visit colonies and mate with who they will.
So let me see. Was there any? Daisy says, "Hi Chuck, thanks for having us."
Daisy. I think Daisy's
Thanks
been
for coming.
following me over on TikTok since about day one, which is I just deeply appreciate Daisy.
Yeah. So I didn't even tell everybody. So I even met. I keep wanting to say Chuck. I met
Yeah, it's okay.
that
Yeah, that's fine.
on TikTok. I mean, I never even thought I would be on TikTok, and then one day I just started making little goofy videos,
Same.
and then I came across your some of your content, and I was like, oh, cool. This is science people. I love science people. And
Science people. Yeah, they're
when I
my
saw
people.
when I saw you specialize in evolutionary biology, I was like, oh, I gotta ask if he can come on.
Yeah,
Um,
too cool.
so I have a question about research. Okay, so there's a lot of bee research that gets done a lot
Yeah.
by a lot of researchers, I guess. But a lot of them are. Let's see. So my question is going to be about bias in research.
Hmm.
So I recently kind of gave my feelings about a research project that happened a few years ago. It went over a few years, and then I think it just ended. But um, how hard is it to remove bias from? So here's an example. I thought of this at work today. Say I want to find out if grass-fed beef is healthier than corn-fed beef. It seems
Yeah.
like a pretty straightforward test. You get a bunch of cows, you feed them grass. You get a bunch, you feed them corn.
Right.
But the problem is, I'm a cattle farmer, and I feed my cows grass, and that's what
Right.
I've been doing for my whole career. Can I effectively direct people to go through this, whatever all the processes are of gathering data and present them without bias? Is it possible? I
Yes.
could be
Yes.
wrong.
No, you're not wrong. It is a challenge. It is a challenge. Now the great thing, one of the things I love about the scientific process, is the way that, and let me first just say that we have to distinguish between like individual observations that you and I may make in our backyard or even of our, you know, our cattle, and they could be really sound observations. Big differences between those kind of observations and us drawing inferences from the results, and us actually prepare. A couple of things: us having gone through the rigorous process of designing an experiment, and a well-designed experiment will always have, sort of first and foremost at its core, a drive to eliminate bias, just simply through the design of the experiment, so that what your feelings about grass-fed beef versus grain-fed beef don't matter, even if you have really strong opinions, right? That's kind of the beauty of the scientific process, is that we go through a number of steps long before we ever put the experiment in place or collect a single data point. We've gone through steps to try to assure that bias doesn't enter into our analysis, or certainly into our experimental design. Then after that, even after we have collected the data, and there are a number of ways, even when you're collecting data, to try to eliminate bias. You know, you can have people collecting the data for you who have no interest or even information about the specific study. There are all sorts of ways. Like, that's just one of literally thousands of ways we can work on eliminating bias at the data collection stage. Maybe not thousands, but lots of ways to eliminate bias at the data collection stage. And then at the results, the analysis results stage, again, we're using mathematical and usually statistical tests that quite literally are designed to address potential bias and either identify it or eliminate it in the analysis. And then finally, and you know, if we're really talking about the way science gets done and disseminated, the information disseminated to people, we're talking about peer-reviewed papers. You know, we're talking about papers that have gone through that whole process that I just talked about, you know, and then before they ever see the light of day, they get absolutely skewered by 3 or 4 reviewers, and it's always reviewer 2 that's the real stick in the mud in my experience. Reviewer 2, man. But yeah, there's always a set of reviewers that are experts in that field who are tearing your study apart, looking for things like bias or error in your interpretation, again, maybe because of your bias. It's a challenge to do backyard science, because, well, backyard science is awesome and we should all be doing backyard science. But we're not really trying to publish our backyard science. So it's important to recognize that while backyard science is vital, it can be beautiful, it gets people interested in not only their backyards, but in science and nature. It is not the scientific process that we use to be able to say something definitively. The only way we can say something definitively, yes, this is true, my hypothesis is true, my hypothesis is false, is really if we've gone through this process that at every step is really designed to eliminate bias, so that we can be as sure as possible that when we say this is true, this is actually happening, that it's true, right? It's not just our interpretation of what's happening. It's our rigorous analysis from start to finish, to say, yes, this is happening, or no, this isn't happening. So
yeah,
well, it's baked into, so eliminating bias is baked into science. But not everything we do scientifically goes through that same rigorous process.
Yeah, I think that, like I was telling you before we went live, that the rift between beekeepers who do the the bee farming standard things that you do, the same thing that the commercial beekeepers do, that group and people like me, it's like so divided. So some of the research that is done is, it'll often be treating bees versus not treating your bees. What will happen? And I can tell you what will happen before anything's done. And I've said, all my people who view me know that I say this often, but it's kind of like saying I got 2 people, one I'm going to give a bunch of steroids, one I'm not, and they're going to work out which one's going to get the most muscles. It's very, very obvious what's going to happen. But for me, I don't really. They're trying to determine best practices for agricultural honey farming and pollination, which means keep those bees alive, which I can understand that treating may help you keep more bees alive. So for over here, my main concern is selecting for survival. I want to enjoy
Right.
the natural bugs, the way they do what they do.
Yeah.
I don't want to become a chemist in my backyard and just really it just doesn't seem fun to me. But that's kind of what this is about. I don't know where the heck I was going with that, but
Well, no, I mean,
so
it's too bad there's such a rift. It's too bad. It's like you either use chemical inputs or you don't, and if you're on one side, the other side, and you kind of vilify the other side. It's kind of like, look, we're all trying to like promote bee survival. We're all
yeah,
trying to sell a product, and like let's just recognize that we're all sort of in this together. You do what you want to do, even if I disagree with the methods, and I'll do what I'm going to do. And if we keep working on it, hopefully sooner or later somebody will stumble on that really dynamite combination of like that really enhances hive survivorship
right.
in the face of a really nasty pest.
Yeah. I
Um,
mean, I used
yeah,
to be
it's too
much
bad it has
worse.
to be so contentious. Like that's
Yeah.
crazy.
I'm getting better. I've
Yeah.
come to I've
So
matured.
it's you. So you're driving it.
It might be me, but it's me. It's that Taylor Swift song.
Yeah,
But
yeah,
yeah, I've kind of matured a little bit to the point where I can say, "Hey, you guys do what you want to do.
yeah,
I'll do what I want to do. We could work together if you wanted to. You could keep them alive and then just have genetics like from people like me. It doesn't have to be me, but I feel like they could benefit from having bees that get their butt kicked
yeah,
and keep going and just use them commercially. But I think it would really mess up the whole system if bees didn't die. Pretty, they have to die in pretty high numbers because from the stories that I hear of the golden years of beekeeping, like no bees died. Like everybody just lived every year, and it doesn't make sense to me because
wow,
we would be sky high in bees if nothing ever died. They have to die. Um, so I
they
really
just
don't even believe that.
yeah. No, that's really interesting. Yeah, really interesting.
We'll see if there's any more questions here. Other than
It
scientific
does.
method.
Yeah, Dr. Time Waster. Awesome. Yeah, and it really does. It's not perfect. Scientific method isn't perfect, but it really is designed, among other things, but to eliminate bias or at least help us identify bias when it creeps in. Um, and yes, that's one of the things I love about it too. I really do, and it's one of the things that I think a lot of people don't recognize about science and the scientific method. Where we live in a crazy time, and science information gets scattershot all over the place, and people are kind of left to pick and choose, and that's problematic. But one thing that I hope does sort of get better communicated is that good science is bias free, and it doesn't mean the human who's doing it is bias free. Um, because we're all human. We can have opinions, we can have biases, but the scientific method, especially when you go through the peer review process, so you're publishing your work, the scientific method is designed to eliminate, at least identify bias and eliminate it where possible. But when you're writing your paper, or after the reviewers have looked at it and identified ways that you need to address potential bias, those are things you can literally address in your manuscript as you're trying to publish it. Um, so yeah, science tries as much as possible to eliminate the bias that is inherent in all human beings. Yeah.
What's funny is I read some kind of studies that were specifically about bias. Like, how do you do a study about bias
Yeah,
and not be biased while you're doing? Like,
that's interesting.
it gets really really deep sometimes.
Mhm.
Trying to remove bias. Sometimes it's just someone might not even really realize that they're biased in certain ways.
Exactly.
So you can do like double blind
Mhm.
studies, but I don't know if that always works. I'll take a couple more questions and then we're right at an hour. So I
Yeah,
think I got every question that I had out of the way, and I just had a great time talking. I'm
yeah,
sure there'll be a
it's
lot of
truly me
follow
too.
ups. We could do. I know you're growing your YouTube channel over there a little bit, and maybe you'll eventually do
Trying.
one.
Yeah,
So anybody
trying.
watching, go check out uh, Chuck. I should not forget your name. Chuck Darwin. God damn. I mean, Charles Darwin, Chuck Darwin.
Chuck, old Chuck Darwin.
It's hard to blindly test things out on bees. You want them to survive, but that affects the results and taints the test. I would say, yeah, you want your bees to survive, and trying things out keeps them alive. But as Seth was saying, some organisms otherwise would not be alive if you didn't keep them alive. So it's kind of just, in my opinion, I might make some people mad, but it's just setting you up for failure down the road. Great to see you. Dr. Timemeister loves your microscope TikToks.
Yeah. Thank you so much. That means a lot to me. Your support is much appreciated.
All right. So I think we went over everything I wanted to. I just want to thank you again for coming on. It was a lot of fun. And yeah, let me know if you ever want to come back on again. We'll after you learn, you'll probably want to learn a little bit about bees now. I mean,
I'm on my way to go learn more about bees and beekeeping. And yes, let's do this again for sure.
Cool. All right. Thank you.
Yeah,
to everyone who's watching, thanks everybody. Have a good night, and we'll see you guys on the next one.
Good night.